The 5 kingdom classification that we study, it easily puts the prokaryotic cells in another kingdom which is totally different from others, and that kingdom is known as kingdom Monera. We did talk about that in the introduction. Now what is kingdom Monera all about? Simplest way to say what kingdom Monera is all about, it is all about bacteria. So basically we are going to deal with tiny microscopic prokaryotic cells. What are the dynamics because of which these cells work, we don't get into that because that is part of our cytological knowledge. We have to study the classification for once. Now what is the classification? How are these bacteria classified? Now had you been students of lower classes, one thing that would have come to your mind is what is bacteria? Anything which causes diseases. They are germs basically. But when we come to this level of study, we know that bacteria has different levels of classifications. What are those levels? We are going to study. Apart from that, bacteria have a lot of they have ecological importance, but I was to say economic importance. They have a lot of economic importance, both positive and negative, like the curd that you make at your home is made up of bacteria, and the heavy bills that you pay at doctors' clinics, that is also because of few or other bacteria. So we are going to see the bacteria in detail. What is the various criteria? What are the various criteria according to which bacteria are classified? Further classifications we have to see. And apart from that, because we cannot just ignore the phylogenetic systems and their considerations, we are going to study a broader classification. There is a classification of kingdom Monera in itself, that is the broader classification of putting few bacteria under one class and the other one under one class, and that classification you need to keep in mind, that is to be remembered. One is Archaebacteria and other one is Eubacteria. Now over here also, keep the bacteria part aside and see what is hidden in the words. What is hidden in the case of first classification, that is Archaebacteria. We use the word archae for something which is old, which is historical, like we have archaeological sites, archaeological evidences, excavations, those are for sites, historical sites are being dealt with over there. Over here, the bacteria which is historical, that means those bacteria which is highly primitive and are supposed to be present since those times when earth was in its formative years, that is Archaebacteria. Alright, one thing that comes to mind. Other one is Eubacteria. Keep the bacteria word aside, then you have true bacteria. So whatever characteristics which we are going to study in detail would imply for Eubacteria, not for Archaebacteria. In the Eubacteria, we have cyanobacteria and other heterotrophic bacteria. We have the classification units and classification entities applied on these Eubacteria. We are going to come to this classification, but before that, let's get into the simplest one, that how the bacteria would be classified. The first classification that we do is on the basis of shape. On the basis of shape that the bacteria has, we have 4 types of bacteria. This you have done in your childhood classes also, so I am just going to recall the terms. One is bacillus, that means rod shaped. Second is coccus, that means spherical. Third is vibrio, that is comma shaped, and last is spirillum, that is spiral shaped. So these are the 4 shapes that we have. Bacillus would be something like rod shaped, coccus would be spherical, vibrio would be somewhat comma shaped, and spirillum would be a spiral structure. This is what the simplest shapes would represent. Now we have diplobacillus if there are 2 bacillus cells. If we have coccus, diplococcus, streptococcus, many cells are arranged in a common form. So basically what a bacteria is, it is a bacteria, it is a cell, small cell, prokaryotic unicellular organism which could be arranged in following arrangements according to the shape, and these would be the classifications. Now, second classification that we have is on the mode of respiration. Alright. We have two modes of respiration. One is aerobic and other is anaerobic. So, the bacteria would be aerobic, that is dependent upon oxygen, and the second one would be anaerobic. Alright. Now, the aerobic bacteria would fall into the class of eubacteria. They will not be in the case of archaebacteria, because I told you that archaebacteria are specialized to survive in those places which have quite hostile conditions. Like I told you that they are present since the early earth years, that means when the earth was in its formative stage. And you have to remember one thing, that earth during its formation was not oxidative. It was in the beginning, it had reducing atmosphere. So, the bacteria which are anaerobic, they would be mostly archaebacteria. That means they do not need oxygen for their survival, and the aerobic bacteria would be dependent upon oxygen for their survival, of course. Then on the basis of nutrition, you see, it is such a diverse kingdom. Also, you have to remember that these bacteria, they have lot of division capability. They are far more advanced, you can say. They are far more advanced in their divisions. That means they would divide at a very rampant rate if we compare it with the eukaryotic cells. So, there is mode of nutrition, that is the third classification criteria. What is mode of nutrition? Mode of nutrition would either be autotrophic or it would be heterotrophic. And because we say that most of the bacteria, they cause diseases, so how can we skip parasitic? Okay. Now, in the case of autotrophic bacteria, we have eubacterias, cyanobacteria. Earlier it was known as blue-green algae, but later on it came out that it is a prokaryotic cell. So, we have cyanobacteria which have heterocysts present in them, responsible for nitrogen fixation, and at the same time they are photosynthetic in nature. That means they are dependent upon sunlight for making up their food. Then we have heterotrophic bacteria and parasitic bacteria, which could be either eubacteria or they could be archaebacteria. So, this is what we have in the class of classification of bacteria, that there is on the basis of shape, on the basis of respiration mode, on the basis of nutrition mode, we have these kinds of bacteria. Then we have another classification for the eubacteria. I am not talking about archaebacteria, basically it is about eubacteria, that whether the bacteria is gram positive or gram negative. Now, this classification lies for archaebacteria and eubacteria. We are going to study about them in detail, their characteristic features, but for now we are seeing the classification criteria. What could all they be? They could all be gram positive, gram negative. This is a classification which is made up on the basis of gram staining. There was a scientist known as Christian Gram. He gave this gram staining technique which distinguished the gram positive and gram negative bacteria. Now, this difference that comes, that the classification difference that we arrive to, that is on the basis of peptidoglycan. Peptidoglycan is a compound that is present in the cell wall, or rather cell envelope of the bacteria. These gram positive bacteria, they have thick peptidoglycan layer, but the gram negative bacteria, they do not have that thick peptidoglycan layer. There is this peptidoglycan layer present between the three layers of cell envelope, and that is not thick. These gram positive, they would have this in abundance, and that because of that they would show a peculiar behavior in the gram staining. Now, what is that peculiar behavior? These cells, they retain, or these bacteria, they retain crystal violet color even after counter staining with safranin. That means they would stay violet. The gram staining process basically involves the crystal violet stain that is added. The bacteria are given the crystal violet stain first of all, then iodine is added, which makes sure that the peptidoglycan imbibes this crystal violet completely inside it. Now, when this peptidoglycan would be in abundance, those crystal violet would be controlled, or they would be trapped inside the layer, that is the cell envelope of the bacteria. But if this peptidoglycan is lesser, the crystal violet would not be trapped completely. When the iodine is added, they cannot completely trap the crystal violet, and then there is a counter staining process by safranin. These, because they have lots of crystal violet trapped in their peptidoglycan, they will not take up safranin, but the gram negative bacteria, they would take up safranin and they would appear pinkish. So these stay violet, and the gram negative bacteria, they stay pinkish after counter staining with the entire gram staining process. Counter staining is done by safranin, and this counter staining is done after the bacteria has been washed with alcohol or acetone. So let us see what are the steps of gram staining once again all over. We know that the basis would be a peptidoglycan layer that would distinguish between a gram positive and gram negative U bacteria. Let us recall what are the steps, and in the next lesson we are going to see the differences that a gram positive and gram negative bacteria has. First of all, the bacteria is heated, a little bit, a smear of bacteria is made. So the first step is smearing of bacteria. The second step would be washing with crystal violet. All right. The third step would be adding iodine. Fourth step would be washing with acetone, alcohol or acetone, and last would be counter staining with safranin. Now you know safranin is pinkish in color. The bacteria that would have lesser peptidoglycan would appear pinkish, while the one which has greater, which has imbibed violet, crystal violet completely due to iodine, that would have the violet coloration. So that is the difference between gram positive and gram negative bacteria. These are the various classification criteria that we use for the classification of bacteria, because bacteria are numerous. And from the Whittaker's point of classification, we have only Archaebacteria and Eubacteria. In these two further classes, there are different modes and different classification bases where the bacteria can be classified. Next, we are going to deal with the gram staining differences and the economic importance that the bacteria holds in the next lesson.